Hello everyone, welcome to my channel VRS Pharma tutorial. My channel is completely dedicated to pharmaceutical analysis. In this video, I am going to explain about basic introduction of pharmaceutical analysis, which involves definition, scope, types and techniques of pharmaceutical analysis. Pharmaceutical analysis. As name itself mentions that analysis means analyzing of the substances. So the definition will be in this way. Pharmaceutical analysis is a branch of pharmaceutical chemistry which involves process of identification, determination, quantification and purification of substances. Here, the qualitative and quantitative determinations of substances are done by manual, chemical, and instrumental methods. There are three types in pharmaceutical analysis where qualitative analysis, quantitative analysis, and semi quantitative analysis. Qualitative analysis. So, qualitative analysis, as I told before, it is a type of pharmaceutical analysis. So, it involves in identification and purification of the substance. Okay. Substances are classified and identified based on physical and chemical properties. So, physical properties like molecular weight, solubility, and chemical properties like reactivity and mass spectra. Okay. So, qualitative analysis, we can give an example of functional group. The substance which contain a functional group. The functional group we can identify, but we can't quantify the substance amount of substance present. So the functional group can be identified in a given sample. Quantitative analysis is another type of analysis. Okay, here we can determine and also quantify the substance. So quantification means the amount that of a sample that is present. For example, the amount of solute present in the solution. Such a way we can quantify it in its own units. Quantitative analysis is the analysis of amount or concentration of an analyte that may be expressed or determined or estimated as the numerical value in the given appropriate units. As I given an example before, concentration of solute that present in the solution. Semi quantitative analysis. This may be considered as a type or may not, but it involves approximate concentration of element in sample. That means it gives a range whether it is below the limit standard limit or above the standard limit. So, for example, determination of impurity present in sample is below or above the specified limits, like limit test what we perform in PIC. Techniques of analysis. Basically, there are two types of techniques in analysis, instrumental and non instrumental. In instrumental analysis, we involve instrumentations. So, we use instrumentation for the purpose of analysis of the substances. Coming to non instrumental, we are not using any of the instruments here for the analysis. Techniques of analysis, as I told before, instrumental and non-instrumental, right? So instrumental, in instrumental there are further classification like electroanalytical methods, spectrometric analysis and chromatographic methods. So in electroanalytical methods, potentiometry, conductometry and difference are there. And spectrometric analysis, as name itself indicate, spectroscopy. So spectrums, okay, atomic absorption spectroscopy, infrared spectroscopy, UV spectroscopy, mass spectroscopy, and atomic emission spectroscopy. Coming to chromatographic methods, TLC, gas chromatography, paper chromatography, high performance liquid chromatography, affinity chromatography, ion exchange chromatography, column chromatography, adsorption chromatography, high performance thin layer chromatography, UPLC methods, and etc. Here I mentioned ELISA, which comes under RIA, that is radioimmunoassay, and ELISA means enzyme linked immunosorbate assay. Coming to non instrumental analysis, technique are also classified into two types, chemical and biological. In biological methods, we involve living organisms like microorganisms for the analysis. Coming to chemical, we have two types, volumetric and gravimetric. Gravimetric analysis is again further divided or classified into direct and indirect. Okay, so in volumetric analysis we have different types of titrations, where volumetric analysis is also known as titrimetric. Okay, so acid base titration, where we go with the reaction of acid and base, redox titration, complexometry, precipitation titrations, potentiometric titration, and non-aqueous titrations. Till now, we came across introduction, techniques, and types. Okay, so now we shall know about scope. So, as we know that pharmaceutical analysis plays an major role in maintaining purity, safety, efficacy of the drug. So, the scope of analysis is spread among the fields. So, coming to quality control, here the pharmaceutical analysis is much more essential to conclude that the pharmaceutical product is stable, and the uniformity, potency, and purity will meet the quality specifications before entering the market. Okay, then drug development. Coming to drug development, the pharmaceutical analysis, it helps in developing a new drug by identification and characterization of active pharmaceutical ingredients and excipients. Coming to method development and validation, here analysis helps to develop, validate, and optimize analytical methods to ensure the accuracy, reliability, and precision of the data. Coming to pharmacokinetics and bioanalysis. So, here the analysis of how drugs are absorbed, distributed, metabolized, and excreted from the biological system. Stability testing. So, here the analysis is very important for analyzing the shelf life of the product and to recommend the storage condition for the product. Impurity profiling. In impurity profiling, to identify, quantify, and characterization of the impurities which are present in pharmaceutical product to ensure their safety and efficacy, it is required. So, in other fields like food determination, biological sample determination, dairy products, forensics, soil study, diagnosis, and farming, etc., the pharmaceutical analysis is much more essential. Thank you for watching the video.